In this lesson, we'll look at the role of semen in fertilization. In particular, we'll focus on some of the research by Spallanzani in the late 1700s, and this is the same Spallanzani who did work on spontaneous generation. Now, it was long known that the female egg was instrumental in the reproductive process. After all, many animals hatch out of an egg. The question was, what role did males play? It was known that males do produce a fluid called semen, but what did semen do? Was it even involved in reproduction? Progress on that question wasn't made until the invention of microscopes. Now, one of the early users was Leeuwenhoek, and he was commissioned by the Royal Society to examine human semen. And he examined his own semen using a microscope like this. This is a very small microscope. The lens is right here, and he would put whatever specimen he was looking at on this tip here, and he could control it using these screws. He could control the focus. Well, when he examined his own semen, he found something startling, and we'll take a look here. He saw lots of little swimming things, almost as if they were animals themselves. So semen was composed of a fluid, but within that fluid were these little swimming things. And for now, we're going to call them spermatic worms. By the late 1700s, Spallanzani is going to pick up the question of the role of semen, and he's going to use frogs as a research subject. Now let's just remind ourselves how frogs reproduce. So they reproduce in the water, so the females lay eggs and the males deposit semen on the eggs. The eggs hatch into tadpoles, and that tadpoles go through a metamorphosis into the adult body plan. Here we see the picture of the mating process. The female has laid her eggs and the male is depositing semen. And Spallanzani knew at this point that semen contained a fluid and also these swimming creatures, or spermatic worms, as Leeuwenhoek had observed. We'll just note here that frogs are engaging in external fertilization, where the fertilization of the egg is happening outside the body. And what we mean by fertilization at this point is that the egg is triggered to begin development. So Spallanzani is going to use the frog here as a research subject to study the role of male semen in the reproductive process. So three questions presented themselves. Was semen even necessary for fertilization? Did semen carry some kind of vital force? And which part of the semen had fertilizing power, the fluid or the spermatic worms, or possibly both? For the first question, was semen necessary, Spallanzani did a very simple thing, but it took some ingenuity. He wanted to block the semen from getting to the eggs, and he chose to fashion little trousers for the males. And he wanted to do this because he wanted the males to still engage in their reproductive behavior, perhaps triggering the female to lay the eggs, but simply physically block the semen from getting to the eggs. And when he did that, he found that the eggs do not fertilize. They do not develop. So from this observation, Spallanzani concluded that semen was necessary for fertilization. And now on to the second question. Did semen carry a vital force? Here again, Spallanzani does something clever. He takes some unfertilized frog eggs and wraps a thread, a fine thread around the eggs so that he can suspend the eggs over some semen. Now this is air in between here. And he reasons that perhaps if semen had some kind of vital force, perhaps that force could act at a distance and trigger the eggs to develop, to fertilize them from a distance. Well, he suspends them over the semen for a little while, takes half the eggs, puts them in fresh water, half the eggs into fresh semen, and the results were clear. The eggs in the water did not develop, the eggs in the semen did develop. From this, Spallanzani concluded that physical contact is required. There is no vital force. And now on to the third question. Which part of semen had fertilizing power? Was it the little swimming worms, the spermatic worms, or was it the fluid component, or was it both? Here Spallanzani took advantage of something he noticed on the microscope. When he would put a drop of semen on the microscope slide, he noticed that the worms tended to gather around the edge of the drop, thus creating a clear fluid area in the middle. So he thought if he could just suck up some of that clear fluid, then he could test whether the fluid itself had the power to fertilize. Notice his strategy here. If you want to know whether the worms or the fluid have the power, you need to separate the two. and test each one. So he sucks up with a little pipette some clear fluid and deposits them on some unfertilized eggs, and what do you know, they develop. So at this point, Spallanzani is going to conclude that the fluid has fertilizing power. Now, you might be suspicious of this conclusion. We can see how he arrived at the conclusion, but we can also criticize the technique. After all, as he sucked up some of the clear fluid, he might have also, unbeknownst to him, inadvertently picked up some of the worms as well. Nevertheless, at this stage, Spallanzani is concluding that the fluid has the power. Spallanzani went on to do a filtering experiment, again to see which part of the semen had fertilizing power. Now this is a good design. What he's going to do is take a filter paper here and pass some semen through the filter paper and collect the fluid down here called the filtrate, and then use that filtrate and apply it to some unfertilized eggs. So the filter paper has tiny little holes, but the idea was to trap the little spermatic worms in the filter paper. Now let's take a look at his results. When you use one filter paper, this filtrate, this fluid down here, can fertilize 100% of the eggs in the dish. It has great fertilizing power. Now, he went on then to add another filter paper, so 2 filter papers, and then 3, and then 4, and you can see his results here. When you use 2 papers, 80% of the eggs fertilize. When you use 3, 60%, 4, 40%, 5, 20, 6, 10. And when he used 7 filter papers, passing semen through 7 filter papers, the filtrate had no power to fertilize the eggs. Now these numbers are sort of made up here, but they capture the actual trend in his data. Now you might explain this by saying, well, as you use more and more filter papers, more and more of the worms will be trapped. So if the filtrate is losing power as you add more and more filter papers, that suggests that it's the worms that have fertilizing power. Surprisingly, Spallanzani concluded the opposite. Spallanzani concluded that the fluid has fertilizing power. Now, why in the world would he conclude this? This is something of a puzzle to historians, but from a psychological point of view, you'll recall that he already was thinking that the fluid had the power from his first experiment with the microscope slide. And so historians think this was his reasoning. He had to agree that the filtrate loses power with more and more papers. However, Spallanzani thought the fluid on the filter paper still retained the fertilizing power. Now, why would he think that? Because when he took those 7 filter papers and squeezed them over top of some unfertilized eggs, they developed. And what he thought was happening was that the fluid loses power as it passes through the filter paper, but the fluid retains power if it is still on the fluid paper. And by squeezing the filter paper, you can get that powerful fluid onto the eggs and they will develop. Now again, we can be critical, because when he was squeezing those 7 filter papers, it's quite possible, in fact very likely, that some of the spermatic worms got squeezed out as well. So Spallanzani, from our point of view, he made some errors. He was perhaps a little careless, or we can criticize his experimental techniques. He arrived at the wrong conclusion. Spallanzani thought the fluid had the power. Today we know it is the spermatic worms that have the power. In the 1800s, with better microscopes, researchers actually observed spermatic worms entering the egg. In fact, when one spermatic worm enters an egg, as you see in these pictures here, the egg will form this fertilization membrane to prevent other sperm from entering. And so, in time, in the 1800s, the scientific community became convinced that these little spermatic worms, which we call sperm cells today, were genuine sex cells. These are male sex cells. So females are producing egg cells, and the semen, that fluid with these swimming things, the semen is the male sexual fluid. The actual sex cell involved are the sperm cells. And of course, later researchers would identify a nucleus inside each of these sperm cells. Of course, the egg had a nucleus, and so both mother and father are contributing genetic material to make the offspring of the next generation.